looks like 5 really nice talks, undergraduate talks. So I'm Ian Schneider. I'm a faculty member in chemical engineering, but I also have a courtesy appointment in genetics development cell biology. And so our first talk is by William Crow. He's both a biology and mathematics major. His mentor is Clark Kaufman. The title of his talk is Locating the MRP ruby trans primordial germ cell reporter line in Drosophila. So I will go ahead and do that and hand it over to Will.
All right. Hello. My name, as you said, my name is William Crow, and I'm a senior here at Iowa State. For the last 4 years or so, I've been working with Dr. Clark Kaufman studying the migration of primordial germ cells in fruit flies. And so, as a little background on what primordial germ cells are, I'm going to play these 2 movies. So as you can see, this clump of brown cells right here they are moving throughout the embryo as it develops, and so those are actually the primordial germ cells. And if I speed things up a little, actually, okay, yeah. So yeah, if you look at, they're starting to coalesce into clumps. Which, if you look at this image down here at the bottom, you can see these clumps here are going to end up becoming the gonads, and so these cells will eventually become the sperm and eggs in the adult flies. And so, a very important group of cells, and we're interested in how these cells migrate and the factors that control them throughout this migration. And so, we're looking at ways to better visualize these cells, because the images you're seeing here, both in the movies, are actually fixed embryos, which means that the embryos have been killed and then they've been stained to identify these cells. And that means you can't actually watch these cells migrate. You can only look at it at specific snapshots that you see down below. And so what we want to do is actually be able to watch these cells migrate in a live embryo to better understand the mechanisms of that migration. And so the way we're doing that is by using fluorescence. And so you see here, we have fluorescent cells that are expressing that MRFP ruby gene. And so that gene creates a red fluorescent protein that, when exposed to a certain wavelength of light, actually produces this red fluorescence. And so, in order to get this image, we actually inserted that MRFP ruby gene right into the fly genome, as you can see in that figure over there, and along with the green, represented in green, that plasmid vector. And so we actually have that gene inserted into the fly genome using transposable elements. And so several researchers a few years before I joined the lab performed those experiments and generated multiple different fly lines that have inserts of that fluorescent gene. And so an issue with those fly lines generated by those previous researchers was that this fluorescence wasn't showing up early enough. So if you see this clump of cells right here, looks a lot like a clump of cells. And as you can see, the migration's already almost complete at that point. And so what we would like to see, we would like to be able to get that red fluorescent protein to show up at about 4 or 5 hours, just so that we can actually watch them migrate throughout the embryo. And so the technique that we've been trying to do is to basically increase the copy number. So as I said here, in this slide, we insert only 1 copy of that gene. And so what we're trying to do is basically increase the number of copies of that gene, and hopefully, which will hopefully make increase the expression earlier in migration. And so in order to do that, we selected the 2 brightest lines that were created in that those initial lines. And then we're going to do a series of crosses that utilizes meiotic recombination to actually get 2 copies on the same chromosome. So if you, the technique we used to insert with transposable elements actually puts that insert in a random spot in the genome. And so if we can get them on two flies that have the insert at different locations, when you cross them, you can actually, every once in a while, meiotic recombination occurs and we'll actually flip one copy of that gene onto the other chromosome. And so then you get one chromosome that has two copies as opposed to one. And so and then from that we can get a fly with 4 copies as opposed to the original 2. And so we're hoping that double the expression will actually increase our visualization earlier in the migration. And so this is the project that I've been working on for the last several years of trying to locate these inserts and then actually do some of these crosses trying to create this fly line. So as I mentioned before, the issue with the transposable elements is that these inserts are going into random spots in the genome. And so we actually can't screen. We can't find this fly line out of all of the ones we create without being able to know exactly where to look for these 2 inserts. And so what I've been doing is I've been trying to find the initial location of those 2 spots so I can screen future fly lines for that insert. And so I've been using a technique called inverse PCR to do that. And PCR, or polymerase chain reaction, is basically a technique used to replicate a piece of DNA. The only issue is you have to know the sequence at both ends of it. And because my insert was put into a random spot in the genome, I actually don't know the 2 ends on either side, represented here by the green lines right there, the green arrows. And so we use a trick called inverse PCR where we actually cut the DNA at those red hash marks, and then you can actually circularize it, which allows us to actually use our known DNA from my insert and then open up that circle again, and actually then we can get 2 known ends down here in the bottom using the actual insert, and then that blue part in the middle will be that unknown region, so we can actually replicate that. And so the results of that experiment are seen right here. So these are the results of the 2 different fly lines that we chose that had the brightest expressions. We had 1-3 and 1-7. So what you're seeing here is from a gel electrophoresis, and we're basically looking at this just visualizing that replicated band. And then using a process called gel extraction, we can actually remove the DNA from this gel and then send it to our sequencing facility here at Iowa State, and they'll actually send me back the DNA sequence of these pieces of DNA. And so one great thing about working in fruit flies is that we actually have public access to the entire sequence genome, and that actually allows us to compare the sequence I get back from the sequencing facility to this public domain genome and actually locate where this blue segment you see here is within the fruit fly genome. And so in order to confirm that location, I ran another PCR, but this time, rather than looking at across the entire that entire known piece, I was trying to confirm the 2 ends using that unknown genome. And so what you see here is the green is that sequencing data that we got from the sequencing facility. And so that was previously unknown sequence, but the sequencing facility gave that to us. And so using that, one of our known ends in there and then one in our insert can allow us to confirm that side. And then if we went to the other side of our known insert and the same thing there, we can get 2 overlapping ends and basically confirm that this 1-7 insert is in this specific location in the genome, which happens to be on the 3rd chromosome. So that's what this gel is showing. The upstream end is going to be the left one, and the downstream end is going to be the right one. So you're just looking between the 2 red arrows. And so I was able to confirm the location of the 1-7, and then I'm currently working on confirming the location of the 1-3 insert. This gel looks similar to this 1-7 gel, but it actually contains very different information. I'm actually showing here, rather than looking at both overlapping ends, I'm looking at the two farthest outside primers right here and basically seeing if I can make a product. And so I have a control fly and I have the 1-3 fly. And so in the control fly, you can see there's a strong band there because there is no insert in that fly. And the insert that we are using is actually too large to be replicated by PCR. And so in the 1-3 fly, when there's an inserted piece of DNA between those two known ends, you actually do not get any product because you actually cannot cross that entire insert. And so this is actually very promising results. My next step in the 1-3 line will be to confirm the location in a similar manner as I did for the 1-7. So look at both of those known ends. And then once I know the location from both of these, we can go on and do these fly crosses that I diagrammed out here and actually screen and try and create this individual. You can actually test whether or not they have those 2 inches. And so last spring before COVID, we actually started some of these crosses ahead of time so that they would be ready to screen when I confirm these locations. But sadly, due to COVID, we had to shut down our lab for a few months and we had to toss out these lines. But while creating these lines, we did see some promising signs that we actually were going to get, it actually was possible. So this image was taken by my PI. It shows a white-eyed fly. And so we actually utilized a clever trick with fruit flies along with that fluorescent protein gene, we included in our insert, we included a gene that makes the fly's eyes red. And so what you see here is a fly that's actually lost both copies of that fluorescent gene. And so in the case when it flips over on the other chromosome, if you keep this bottom one, the fly that has neither. And so that's why you see a white-eyed fly here. And so we know that recombination did occur, but we got the other end of it. And so this fly wasn't actually usable by us, but we know it's promising to actually see that it's possible. And so it's a good sign. The only issue was that we only saw 1 out of every 200 flies that had this white-eyed phenotype, which suggests that these genes are actually very close together, which means it's very unlikely for that crossing over to occur. And so in the future, we might look at a different method for basically confirming that location, or a different method for increasing expression of those of that red fluorescent protein, just because it's looking like these inserts might be too close together to actually be a viable option for us. So some of my future directions right now are to finally confirm that location of the 1-3 insert, hopefully reduce some of those crosses. Again, I'm a senior, so I'm going to be graduating in a few weeks, and so that might be on a future researcher next year, hopefully. And then possibly look into alternative techniques to increase the expression of that red fluorescent protein to allow us to visualize those migrating cells earlier. And then I'd like to wrap up with some acknowledgements. Thanks to Dr. Clark Kaufman for mentoring me for the last 4 years, and then also to the College of Liberal Arts and Sciences for funding some of my research. But yeah, let me know if you guys have any questions. Thank you for listening to my presentation. So the top of the genome, like I said, it looks like both of these are on chromosome 3, and I think that's about 32 million base pairs long, and our insert's only about 10,000 or 12,000. So it doesn't really affect. The only issue would have been if it got inserted into the middle of a very important gene, but because these fly lines have been around for almost 10 years now and they haven't had any issue, and so there hasn't been any issue so far of affecting the viability of these flies. Any other questions? So yeah, that's we're looking at a few, but so like I said, previous researchers had actually made these lines, and they actually made them before CRISPR was a really big thing, and so there might actually, nowadays CRISPR has opened up a lot of doors, and so in the future we might be able to look at using CRISPR to insert another copy of that gene as well. But what I've been focusing on is looking at that recombination, and so where it would, if we go forward with that, it would just be screening through about 100 fly lines and hoping to find one or two that are viable. Yeah, correct. So yeah. If I go back to here, if you look, so basically, as the farther apart a gene is, the more likely recombination is to occur between them for the most part. And so it's kind of like if you're throwing a dart at a dartboard or whatever, you're more likely, the farther apart you are, the more likely you are actually, or like the closer you are to the dartboard, you're more likely you're hit. And so similar, okay, that was a bad example. The bigger the dartboard is, the more likely you are to hit it. And so it's kind of that same thing of the bigger the distance between these genes, the more likely it is to occur. And so if I was to draw this accurately, represent these sizes accurately, my 1-3, based on the results right here, this location here, my 1-3 would probably be about 2. And so they're very close together. And so other experiments have been done looking at different spots in the Drosophila genome, looking at how often it occurs between these two locations. And based on those two locations, it's about 1 in 200 odds. And so, and that's just with these two lines. And so, like I mentioned before, they made multiple of these lines, but we wanted to look at the two brightest ones. And so if we had chosen one that was slightly dimmer, that insert might have been here, and it would have been much higher chance of that recombination occurring. And so it just so happens that the two that we chose were very close together.
Mhm. All right, great. Well, our second speaker is Kit Claiborne, excuse me, from animal ecology and biology. His mentor is Philip, psychologist from the Veterinary Diagnostic Lab. The title of his talk is Effect of Freeze-Thaw Cycles on Porcine Reproductive and Respiratory Syndrome Virus, RT-PCR Cycle Threshold Values. So join me in.
Okay, hello everyone. Like he said, my name is Kit Claiborne. I'm a freshman in animal ecology biology, and my mentor is Philip Gauger of the Veterinary Diagnostic Lab in the Department of Veterinary Diagnostic and Preventative Medicine. And I will talk to you today about the effect of freeze-thaw cycles on porcine reproductive and respiratory syndrome virus and the PCR cycle threshold values. So porcine reproductive and respiratory syndrome virus, also known as PRRS. What is it? It's a respiratory disease that also results in the reproduction failure of pigs. And the morbidity rate, which is the rate of the pig actually having the virus, is 50 to 100%. And the mortality rate of that 50 to 100% is 10 to 50%. So obviously, as this shows, it is a very devastating virus. And with this, swine farms have to routinely monitor for PRRS. And this monitoring is the real-time PCR testing, and it is every month or week. And then this monitoring is necessary for the virus and disease control inside of the farms. So the actual PRRS testing, approximately $664 million per year is affected by PRRS related in the swine industry. And these expenses are loss of production and the actual treatment it costs to treat the pigs. And in the year 2020, the Veterinary Diagnostic Lab performed
505,357 molecular tests. And of that number, 198,200 were the PCR testing in just for PERS. So this equates to approximately 39% of all the PCR tests at the VDL are PERS, and it is the most tested virus at the VDL. So in the year 2020, clients spent around $5 million just on the PCR PERS itself. And these results must be accurate, timely, and repeatable to actually control the disease. So we can go into the statement of the problem. PERS positive results may need confirmation. So is this, did we get a false positive from the actual testing? And then for the PCR PERS CT values or the cycle threshold values, we can see that at elevated levels of 35 to 40 actually indicate a low concentration of that target virus. And at these elevated CT levels, there is a poor repeatability in that actual PCR testing. And if we do get a negative retest after the positive, this can imply that there was cross-contamination between samples, or it was just a false positive. And it is also difficult to confirm these weak positive PCR results, so it can cause confusion for diagnosticians, veterinarians, and the clients that actually have the pigs. So our statement or our question that we had was, do freeze-thaw cycles on these PCR positive samples affect the repeatability at elevated CT levels, which also indicate low concentrations of the virus? To determine the actual repeatability, we tested with serum at low concentration of the virus. And then we evaluated these at different CT levels, and all of these samples were stored in a freezer at -80°C. And we had a few questions actually regarding the positive serums. And is there effect of repeatability when the concentrations are low? And what is that variation of the CT levels when retested after freeze-thaw cycle? For what we actually did is we took a 1 to 10 dilution, which is 1 milliliter of positive to 9 milliliters of negative sera. And we mixed that together, and then the rest of the test tubes had 3 milliliters of negative sera, and we took 3 milliliters of the previous dilution into the next tube. So in total, we had 9, and the 2 to 9 were serial 2-fold dilutions, and the first one, like I said, was 1 to 10 dilution. And of all those 9 dilutions, we took a triplicate or 3 aliquots into each PCR machine. So we had 3 of the same dilution in a PCR machine at once. So we had a total of 27 dilutions, and each were around 130 microliters. And then we went through the freeze thaws, which were 0, 1, 2, 5, and 10. 0 was our control, and then that's how we would compare the CT values after the freeze thaw cycles. And 1, 2, and 5 is what we expect clients to actually want to test when they do PCR and see how it is affected by the CT levels. And then 10 is just if there are more freeze-thaw cycles after the clients wanted it, what is the effect of more freeze-thaw cycles? And then each dilution was extracted once by the PCR. And then after we got the results, they were reported as the CT values. And then from there, we took the mean and the variance. And we plotted and we tested all 135 reactions. And there was unfortunately actually no significant difference between each test. So as the graph can see, the X value is actual CT value itself, and the Y is what we expected. So as you get farther into the CT values, there is a little bit of effective of repeatability, and repeatability is how concentrated the results should be. So when we get into the lower CT values, like 29 to 30, there are closer and is more concentrated. But as we elevate more, you can see that there are some sort of freeze-thaw cycles that did not have what we expected. But again, this is only some, so there is no significant differences. And for this, we did, as I previously stated, we took the serum of clinical samples with PCR positive results. And we went through 1, 2, 5, and 10 freeze-thaw cycles, which had little to no effect on the CT values itself. That ranged from 29 to 38. And then regardless of the number of freeze-thaw cycles that we actually did, the qualitative result, which is the CT value, was unaffected. And then elevated CT levels have reduced repeatability, and then the target concentration may have a greater impact on the CT values as a whole. The effect of freeze thaw actually may not have an impact on the repeatability, and these results are only exclusive to serum, which is what we tested. So the quality of that serum that we got from clients may affect the outcome of what we got. And the greater elevated CT values, like I said, they may have a greater impact on repeatability, but in this experiment and this research, there wasn't. And we also have alternative sample types that we could use: tissues, oral fluid, processing fluid, and others. And since these are alternative sample types, there might be a different outcome after the freeze thaw cycles. So this goes into our future research and just future aspects of PCR testing in general. And like I said earlier on the previous slide, we could have taken additional sample types, and these sample types could have been nasal swabs or oral fluid of the pigs. And those two tests are significantly what's going on right now with COVID-19. And when you get your COVID-19 test, specifically at Iowa State, it goes through the PCR machines at the veterinary diagnostic lab. So we could see that there are different variables and different outcomes from different sample types of humans and of pigs on PCR. And with the freeze thaw cycles, we could also say and look at the research what we did and see that if there was freeze thaw cycles for a COVID-19 test, say that they got tested at one place and the test of the samples are at another place, and they have to freeze those samples so they don't get affected. If they did go through a few freeze thaw cycles, is that CT value of that specific COVID-19 test affected? So the PCR machines at Iowa State, once the concentration CT value hits 40, it comes back negative, but there still could be possibly a concentration of slow concentration of virus in that sample. So if there are freeze thaw cycles and it goes through multiple freeze thaw cycles, if it passes that 40, it comes back negative, but that person still could have it, or that pig still could have that virus. So maybe in the future, during another future pandemic or still COVID-19, if multiple freeze thaws affect COVID-19 samples, then we could see, is there a false negative? Does that person still have COVID-19 and still transmitting it to different people, but their actual test came back negative? And then acknowledgements. The VDL, faculty, staff, and students, specifically in the molecular section of the VDL. And then obviously the ISU honors program, that's why I'm here. Any questions? Yes. So our target was around like the 29 to 30 aspect from the beginning. So when we got the clinical samples, we tested the PCR first of the samples that we got without any freeze thaws. And from there, we chose what sample we wanted that would have a CT value of 29 to 30 after the 1 to 10 dilution. So we only took like specifically before we did the 1 to 10 dilution, it was 26. So we only did that for those 29 to 30 dilutions. So we could do other samples and other CT values, but for this experiment and research specifically, we only did the 29 to 38. Yes. Mm-hmm. What we did is we took, we froze them at -80 for like a few hours or a few days, and then we thawed them for 30 minutes at room temperature. And that freeze thaw actually, like, it rips apart the genome. So when there is more freeze thaws, it can affect the genome, which can affect the PCR itself. So I don't know personally if we could, if like 20 years later, it could be if it is frozen for -80 the whole time. But as more samples come, usually at the veterinary diagnostic lab and just labs, they retest and replace. But there could be a possibility that in the future, if there are test results later, then you could come back to it. It just depends on how many freeze-thaw cycles it actually went through. So it depends on how much of the freeze thaw happens because, like I said earlier, when you freeze and thaw a sample multiple times, the genome starts to like break down, specifically in when it gets frozen. So as you go through more freeze thaw cycles, the genome that actually needs to be tested by PCR, because the PCR machine only can detect a certain amount of genome. As it goes through more freeze thaw cycles, the genome breaks down and then the PCR machine can't detect it as it could before. Mhm. Yeah. Mhm. Yeah, so at VDL, the clients usually want to do more than just one freeze thaw, just to see if there is an effect of the CT values. And it also depends on the transport because any transport is different. So most of the things of clients, and specifically at the veterinary diagnostic lab, there are pigs that are there and that are housed there. So it just depends on like the distance between where they got the samples and where they're getting tested. And one freeze thaw most likely won't affect the CT value of any sample. So it might go through one freeze thaw on the way to the actual testing facility, but it shouldn't really have an effect on the CT values as a whole.
Up next.